I'm gonna hit the record button. You don't need to see me, I don't think, while we do this. Oh, maybe. Can I put myself up here? I guess not. All right. Anyway, so first up, I thought it would be good for us to just talk about the virus itself, right? So if you look back to chapter 6, which is literally what I'm doing right at this moment, and there is a picture of animal virus multiplication. So let's start with, that, right? So obviously it's a coronavirus. We all know that, right? And that's the Coronaviridae is the family. The genus is coronavirus, right? And clearly there are currently 3, right, major human pathogens. And I think all 3 of them affect animals as well, right? So there's the original SARS, there's what they call MARS, because it was a Middle East version of coronavirus, and then what we're dealing with now, coronavirus infectious disease 19, right, for the year, for 2019. Now, these guys are all RNA viruses, not DNA viruses, which is why doing the PCR test requires one of 2 adaptations for that to be successful. But first of all, what I wanted to have you look at was the picture on page 153 that talks about, again, from chapter 6, that talks about this particular virus's multiplication cycle, right? So animal virus, so it has to bind, it has to adhere, adsorb, we called it adsorption, penetrate, uncoat, because we need to get rid of those spike proteins and the outer shell of the virus, the envelope. I believe it is an enveloped virus. Then synthesis, remember you have to make viral, in this case, RNA and viral proteins, assembly and release. I did read last night that the virus does form syncytium, so you can actually see that in diagnosis, right? I wanted to write that down, but I never remember how to spell that word, so. Really? It doesn't even say that anymore. Cytopathic effects, right? So we need a syncytium, SYNCYTIUM, form syncytium, and then eventually the cells lyse and die, obviously. So you can actually see that under the microscope if you're looking at infected cells. Now, because it's an RNA virus, and I believe it is sense strand. So I'm pretty sure that once the virus gets in, it can immediately, and again, I'm not positive about this, but I think it's a sense strand, so that as soon as it enters into the cell and gets uncoated, your cells, the host cells, can use the virus as messenger RNA to begin synthesizing virus proteins. Now, something we did not talk about is if the virus comes in and its sense strand or messenger, like, analogous to messenger RNA, if it comes in with that as its genome, do you see the problem where it's going to have to somehow make more of this plus strand of RNA for its genome, right? So the host cells can immediately use the sense strand to synthesize some of the viral proteins, right? Sort of right away, immediately. But we need more copies of this to package up, and we can't make them directly from the starting material. So here's the virus, right? So here's the RNA, and let's just call it a plus strand, right? So when that gets inside your cell, for example, here's your cell, host cell, some of that plus strand obviously can immediately start synthesizing viral proteins. But remember, to make a new virus, we need that, and we need more of these. We need more plus strands of messenger RNA to make new virus, right? How are we going to do that? Usually some of the very first proteins that the virus synthesizes are enzymes that can take the plus strand, convert it into a complementary negative strand that will then be used as template to produce multiple plus strands for genome. And we didn't talk about that when we were in chapter 6, because I told you we're not going to talk about how RNA viruses replicate because it's complicated. And now that we've talked more about DNA, and you've just, you've had more time to chew on that, it can make a little bit more sense to you. Okay, so now hold on to that thought, right? Also, the test that we're using, PCR, what am I doing there? Right, was originally designed as a test to detect rare, low copy number pieces of DNA. Okay, I don't have DNA. I have RNA. This virus has RNA, not DNA. There's no DNA associated with it whatsoever. So this is a problem, because if I want to use PCR to detect RNA, right, and I'm starting with RNA as my starting material, I'm going to have to convert this RNA into a kind of DNA. We call it cDNA, or complementary DNA. C for complementary because, right, it's going to be complementary, obviously. There's an A, right? The DNA, the complementary DNA will have a T. And we first synthesize single-stranded complementary DNA, and then put that in a cell-free system that is going to produce double-stranded DNA, cDNA, from this message. Now, don't freak out, right? So why we call it complementary DNA is because, let me get rid of the share for a minute. If, for example, it was from a eukaryotic cell, there aren't any introns in it, right? So it's just the coding. You can think of it as just the coding region of the DNA, the genes of the DNA. Now, there's a little bit more to it as well. Let's go back to my sharing. I think those are going to be gone, but whatever. It always upsets me, but. So we've got this complementary, or cDNA, right? That is basically the coding messenger RNA, right? So first, and this, we don't want all the DNA. So when we do PCR, it's usually limited to not just a rare or low copy number piece of DNA. What we mean by that, copy number of DNA, a piece of DNA, is it's usually not even a whole gene. Right? Sometimes, but not always. So think about, in terms of this virus, we aren't doing PCR. The test is not testing for all the RNA genome that the virus has. We have to pick. and you pick the one that's going to be best or most frequently expressed, meaning expressed into protein. And so we're going to pick, I think they chose the spike protein. The spike protein is, when you look at those pictures of coronavirus, the spike protein is the picture where it's, you know, it's always gray with the red little like things on this. That's the red thing, is the spike protein. There are, I believe, 3 major proteins that stick out of the envelope, but they chose the spike protein because that's the protein that binds to the ACE2, the ACE2 receptors in humans, and that's how it gets into your cells. So the thought process is, as a scientist, we want to pick something that's going to mutate the least frequently. And usually what that is how the virus gets into the cells, because that mechanism, because if the virus can't get in, it can't multiply. So usually the spike proteins or parts of spike proteins are the targets for these PCR tests. For HIV, it's P24, which is part of the GP120, I think it's part of the big glycoprotein. It's a spike protein that doesn't mutate very often, right? Or very much. So that's why the spike protein is the target. So this is going to be what we call the target sequence. Okay, and I'm just going to put in parentheses here. It's the one that they always show in red in the drawings of COVID-19. Right, they always show it as like this circular thing, right? And then there's these little, they're kind of like in triplets that stick out, these little spike proteins that stick out all over it. So the spike protein is going to be the target sequence. Now, so the steps are, first, you have to collect from a patient, and like Lauren's experience, they're going to give you the lobotomy swab. That's either going to be on a swab where they go back to the deep in your nasopharynx, right? Which is why it's so long and it's a little bit painful, or saliva. You can spit in a cup. Well, it's a little test tube. Right. And so in that, and I was watching a video this morning from someone who was talking about this procedure and how it works. And I thought this was really important to remind students, is that no matter where you're getting your sample from, right, where you're collecting from the human, you are collecting many different types of cells. Right. So you're going to be collecting some of those from BIO1. When you remember looking, you're going to be collecting some epithelial cells, right? Some human sloughed off epithelial cells. You're going to be collecting probably some bacteria, and I'm going to draw them with capsules, because the ones that live in your nasopharynx often have capsules. So I'm going to draw some streptococcus there, and then I'm going to make some staphylococcus, right? So you're going to get some bacteria, make them blue. And we know from the Human Genome Project, right, there's tons of bacteria, though there's just a couple of the more common ones. There are going to be other viruses in there. So I'm going to draw flu because I can. Right, so that's got those 2 spikes, hemagglutinin and H1 and N, H and N, hemagglutinin and neuraminidase. So I'll draw some spikes for that. All right, so this is supposed to be flu virus or any other virus that you might have. Common cold virus, flu, we know has those 9 strands of single-stranded RNA in them, right? Or any other viruses, right? Like rotaviruses, other rhinoviruses, other cold viruses, thing that you might have just picked up, right? So no matter which way you collect, you got all these different cells. And this may or may not have been particularly clear to you guys, but when you are trying to isolate and study which bacteria is making a person sick, I'll say isolate and identify, right? So when you're doing that, you need a pure culture of that bacteria, like you guys had from in your lab kits, right? That's why we made you rehydrate those cultures and then isolate them on petri dishes to try to illustrate to you that if you're going to try to study the biochemistry of a bacteria, you need a pure culture of that bacteria. You can't try to study if the bacteria eats lactose. If you have 4 bacteria in there, one of them might digest lactose and make acids, right? And that's not going to help you figure out which one it is. Excuse me. So this is why it's PCR, because when you do PCR, you only amplify the DNA, or I'm going to put /RNA because we're going to talk about that RNA test. You only amplify the DNA of that target sequence. That's why it's super specific. That's why this works. Not, I'll put that in red, not the DNA of any of these other cells. Exclamation point, underline, 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 right? So that's what makes this makes the PCR so useful and unique to do this test, is because if there's a SARS virus in here, and a coronavirus in here, right? So I'll make it like this, and I'll put the red spikes on it, because you actually have COVID-19. Only one that's going to be amplified by this procedure is this guy. None of the other ones, because they aren't the target sequence of DNA. Okay, so now I'll just briefly remind you how PCR works, and I'm going to use DNA as my example, and then we'll talk about, we've already mentioned how you get the complementary DNA if you're studying for a virus. Okay, so I would have the target sequence. In this case, it would be the target sequence for the spike protein, for part of the spike protein on the surface of COVID-19, right, of that SARS virus. And what we mean by the target sequence is I know every base, right? So if this sequence is A, T, T, G, C, T, A, whatever it is, right, to 3' here, A, T, 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 G, C, whatever. I know this entire sequence. That's what's really important, that I have to know the target sequence. And I'm putting dots because I'm not writing out the whole sequence, the whole target sequence. You can only make this if you already know the whole sequence of the genome of the virus, which, remember, was published like in January. And this target sequence piece, you are going to part of the spike protein. So remember, most proteins, right, they are in the neighborhood of 200, maybe between 1 and 300 amino acids in length, and each amino acid is a codon, and each codon is 3 bases, right? So we're looking at a piece of DNA that we're going to amplify somewhere in the neighborhood. Usually it's in the neighborhood of 400 to 600 base pairs. So meaning we don't even need the whole target sequence for the whole spike protein. We're only going to look at a part of it. We're only going to look at maybe this first 400 bases. Now, originally we would choose 400 to 600 bases because that was what could be visualized really easily on a gel electrophoresis. So we could load the sample after we amplified them and run it on a simple agarose gel, and you could see a band for the pattern, right? Now, okay, lost my train of thought here. Hang on a second. Okay, so I've got the target sequence. Remember, for PCR to work, I need another bunch of things in my, I need this, the target sequence. Now I need to make, meaning manufacture or purchase, primers and they're a set of primers, okay? One of 3' to 5', right? And those primers are specific for complementary to this sequence, 3' to 5', right? And complementary to this sequence, 3', both directions, so that Taq polymerase can just jump on and continue sequencing, making complementary bases really quickly. So I need a set of primers, and then I need lots and lots of nucleotides in a format that can be easily added for the enzyme. So that means I'm going to need A's and T's and G's and C's. And again, you would purchase those in very small quantities and high concentration. And then I need an enzyme that can withstand the heat of PCR, literally, because we're going to heat this reaction up to 95 degrees Celsius. And that enzyme is known as TAQ polymerase. And that's based on the organism that it was originally isolated from. It was a Thermococcus aquatic something organism from the deep sea vents. Okay, but essentially it's a DNA polymerase that works at really high temperatures, has a wide temperature range that it can work at. So we're going to mix all these things together. It's a cell-free system. So I don't need, this makes it speedy, and I don't need to have helicase, gyrase, polymerase 2 or 2, polymerase 1. I don't need primase, right? I don't need an RNA primer because I have the primers already built, and they're going to be DNA primers, so I don't need to replace the RNA with DNA like I would in a normal, if I was just copying DNA. So in this cell-free system, what's going to happen is my super hard to find small piece of that spike protein first, we're going to separate the strands. We're going to break it apart. And what they usually don't tell you about PCR is before you do it, you have to lyse all the cells that are present and you have to clean them up. Right? So you clean them up by putting them over columns that will separate out DNA from RNA and proteins. Sometimes you treat it with proteases to get rid of proteins and DNase to get rid of any pieces of DNA before, in this case, we would be amplifying. You can use RNases too, depending on what you're trying to purify. And then you would make that complementary cDNA, right, from the virus messenger RNA. And then we would be using this cDNA as our sequence. We'd be putting that in as our target sequence. Okay, so that's going in the test tube. Now, the primers are going to bind like this. The TAQ polymerase is going to bind and extend, right? So we're going to heat it up to 95 to separate the strands. Then we're going to cool it down, usually to 60 some degrees. These are Celsius, to allow the primers to anneal. And then we're going to heat it back up, usually in the neighborhood of 72 degrees Celsius for what we call extension. So we'll let those primers extend. And now we are going to literally have 2 copies. Now we're going to have our original target sequence, if it's there, right, from our original cDNA that we cleaned up from our patient sample. This is what we're going to have. Old strand, new strand, old strand, new strand. right? Now we have 2 copies of this. 2 is still too small to see either with quantitative PCR or with just traditional PCR. So we have to make more copies. So we have to do multiple cycles. You'll see that sometimes you'll say we did 30 cycles or we did 50 cycles. So what that means is we repeat 95, 60, 72. And on the repeat, right? So on the next repeat, we still have these guys they're going to get a new complementary strand. And then we also have the complementary strand that we made, and they're going to get a complementary strand, right? So now we have 4 copies, cycle 2. And then we repeat it again and again and again and again and again. And again, now in traditional PCR, you would just look at this then after maybe 30 cycles, you would look at this on an agarose gel and you would just be looking for, over here would be running a standard, and it would be, it's usually 100 base pair ladder. So let's say, I forgot what I said we were going to use for our target sequence here, between 400 and 600 base pairs. So let's say we just used 400 base pairs. So then on our sample, our person 1, if we saw a band at right here at 400, and they're usually like that. And if we, on patient 2, we saw a band, but on patient 3, there was nothing there. It was blank. Okay, these 2 people have the virus, that person doesn't. Done. This is a yes or no. And that's it. That's all you can say. Yes, the virus was present, or no, the virus was not present. What is more detailed information, or sometimes what we say more useful information, is if we could quantitate, right? How much starting material did we have? A little bit or a lot? And it would also be nice if we could see, I'm going to put that in quotations, right? Just like immunoglobulins, you can't see them because they're clear. You can't see DNA or RNA because it's clear. If we could see it in real time or as it's happening. Now, what people confuse is Q is RT. Okay. Actually, no, sorry, let me take that back. So Q PCR, when you see this, that's the quantitative, right? And that is in what is actually happening in real time. And you need a very, very expensive, I should make those green, a very, very expensive, like $50,000 piece of equipment in order to do this. And how that is done is as the DNA is, as I understand it, and again, I've never done this myself personally, but what I was just watching is that the primers have 2 markers on them, 2 chemicals on them, that one of them blocks the expression of the other chemical when they're in a close distance like this. But after the extension, when those 2 compounds get separated further, then one of them glows. It's fluorescence. And then this equipment measures the amount of fluorescence with each cycle. And it puts it on a graph. And you can watch it increase over time. The other type of PCR that you see referred to, RT-PCR, does not stand for real time, right? So that's a little confusing. Q stands for real time PCR. RT stands for reverse transcriptase PCR, which is how you could take a single strand of RNA and reverse transcribe it into a double strand of DNA in one step. This enzyme is an enzyme that has been isolated and purified. That's an ASE from HIV, right? So it's a or a herpes virus, I think it was. So this is getting us to that sort of cDNA in a quicker way. So you might even see sometimes this QRT PCR, right? So if they use reverse transcriptase to get to the double-stranded DNA for the target sequence in the PCR, and they are looking in real time how much that particular sequence is being amplified, you might see all of those letters together, right? So depending on the company and depending on which version of this they're using, that's why you see different types of the PCR. But the premise is still the same. We are looking for a super low copy number, in this case, RNA, and then we are using PCR either at the rapid tests, right? When you find out in 24 hours, chances are they're doing the quantitative one, right? Because it requires less manipulation after the purification steps. It requires less manipulation. Just pop it into a machine, does the PCR and tells you how much. You don't have to collect the sample and then run the agarose gel, which takes like another hour, right? So that's why the testing can be so quick, right? So we've got super low copy number of, in this case, RNA. We're getting PCR, and then you're getting a positive or negative result. And most of us are not on our diagnostic tests. We aren't getting the report about the actual quantity of virus. That would be going to our doctor, that would be going to the CDC, right? But to the individuals, we're not seeing how much virus we individually have. This is helping other scientists try to figure out in combination with contact tracing. This is like everything we've learned in microbiology, right? So this is an epidemiology trick. Right? A tool of epidemiology to see where the pandemic is moving, to see who's infecting who. Now, you can link, like I said, people at the CDC and other scientists are then linking the contact tracing to these quantities of virus present by PCR. And that is helping us figure out, right, is there a number or an amount of virus that would cause someone to be asymptomatic predominantly versus symptomatic, right? How much virus needs to be present in order to be a reservoir, if you want to think of it that way, right? Or to be an actual transmitting person. I don't think that has two T's, right? So someone who could transmit by direct contact, let's say. So all of that, we're not going to know the answers to that until a long time from now, right? And of course, the contact tracing has even more relevance because we're trying to contain the virus. So not behind the scenes, but up front, we want to know who you've been in contact with so that we can try to quarantine those people as well. And because we don't live in a communist country, right, the quarantining is self-quarantining, right? And you have to decide to do it yourself, and that's important to you, and that you actually quarantine. And so far, there haven't been any particular consequences. No one's calling your house to see if you're still there today, right? If we were living in China, I don't know if you guys have seen some of the videos of, it's on people's phones, and then they just track their phone, and if they haven't been quarantining, then they go and put them in quarantine. So here, we're still using self-quarantine, which is not very, it can be effective, but doesn't, in some parts of the country, clearly it's not effective. Most of the parts that are experiencing a big spike in viruses right now. All right, and the last thing that I wanted to talk to us about today was, and then I'll pause it and you guys can ask questions. I wanted to talk a little bit about the vaccine that are being developed. The vaccines, my understanding, all three of them, they are RNA vaccines. So unlike anything that we talked about when we talked about different types of vaccines, remember we talked about whole cell vaccines. We talked about, trying to get to that page in my book because I was like, oh, I put a marker there to remind us. Yeah. So remember, I don't know if you guys remember, but we talked about different types of vaccines, right? Heat killed, whole cell, right? Or whole virus. We talked about attenuated. We talked about what we would call subunit vaccines that are just a piece of a protein or a couple of the proteins from a particular bacteria or virus. So these are even more, right, cutting edge in that we're not even giving people protein anymore in this case. So this is not all three of the vaccines that are in trial and are actually out now. Some people have already, I heard yesterday that people in England are actually already being vaccinated. And I think I think in December in some places in the US, people are, first responders are going to start getting vaccinated. This is just RNA. So it's like tons and tons of messenger RNA that is going to get injected, or I'm not sure, it might be a nasal, right? Like the flu vaccine, it might be a nasal squirt because that's where the cells are that are, it's probably going to be injected. I'm sure there's both versions. And then this RNA for the spike protein for the spike protein. This is going to get taken into your cells. So here's your macrophage, right? And that macrophage is going to take the messenger RNA in and it's going to produce the spike protein. And it's going to put it on its surface in the context of class 2 MHC. Remember that from our immunology talks, right? And then B cells and T cells are going to be able to recognize this. So a B cell could, in theory, also just, let's draw a B cell here, right? A B cell could recognize this protein and be triggered to form plasma cells, but most likely it's going to be this mechanism. A B cell is going to bind to the spike protein presented by a macrophage, and we're going to get B cell clonal activation. And we know that that's going to give us lots and lots of plasma cells that are going to produce immunoglobulins. Yay, right? It's a 2-dose vaccine because remember your first response is going to be high in IgM, low in IgG, and we want the IgG because it's more long lasting. So the second dose is going to trigger all the memory cells, and we're going to get a big spike in IgG, right? And that's all they can measure right now to claim that the vaccines are effective, as well as monitor the people to see if any of them have gotten sick with COVID. Of course, it is also hoped, but I don't know that we know yet if T cells are being activated by this particular mechanism of vaccination. Like I said, I don't know, and I don't know if that's even known, but definitely we're going to get some B cell activation. All right, so I'm going to stop my sharing and stop my recording.